this is a picture, sorry, that we did in our last video. And it's basically a typical looking animal cell and some of the mean organelles that we find inside and outside of the animal cell. There's one thing that I didn't put on this diagram. I thought about it after, so I'll add it now. And that is, there inside the cell there is also a liquid or a fluid. And so I'm going to take one more color here. Maybe I can do it in this, well, I guess I'll just do it in black. And I'll put this little line pointing to sort of nowhere, kind of like just into the empty space. Now there's two words that people use to describe this, and I want to make sure we clarify, because there is a slight clarification, okay? Let's go up a bit. The liquid or fluid inside the cell is called the cytosol. Cytosol, which is basically the liquid that everything is kind of floating in. It's mostly water, of course, with other things dissolved in it. But sometimes you'll see in books the word cytoplasm. And it points sort of in the middle of nowhere, cytoplasm. The cytoplasm actually is a word that refers to everything inside the cell. The entire inner contents of the cell are the cytoplasm. And so the cytoplasm doesn't refer specifically to the fluid. And that's what I wanted to just make sure you understood the difference. Cytosol is the fluid. Cytoplasm is sort of just the word for everything inside the cell. Okay? And they're slightly different words, but people use them interchangeably sometimes. So that's kind of a little bit of a difference. Okay, so now what we're going to do is we're going to go down below here and we're going to start to write little explanations of what each of these structures actually does. And so we'll begin with the cell membrane. Perhaps we did not label that one either. Now it occurs to me, if we go back up here, we labeled everything around it except not the membrane. So again, I'll add one more little arrow. I'll add it in red down here. And that, of course, is pointing to the actual black line here. Right? I'll draw it like that so it doesn't look like we're pointing to the cilia. And this is the cell membrane. The cell membrane is sort of the envelope that holds everything together. So we should add that to our picture as well. Okay, now we're ready to talk about some of the things and what they do. So the cell membrane, we'll start with that. Talk about what it does. All right, so our cell membrane has the following roles. First of all, it forms the boundary that contains the cell. It also controls the movement of things in and out of the cell. Mostly molecules, right? Things in and out of the cell. Of course, I'm going to add that it does this primarily through a couple of different processes. Diffusion, osmosis, which we're going to learn about, and what we call transport. And the transport part is the one where they use the protein gates. Remember the integral protein that can form gateways. That's the transport part. And when we're talking about the cell membrane, there's a word that we use, of course. We call the cell membrane selectively permeable. And so what that means, permeable means how well things can move in and out. So if you're very permeable, everything sort of sloshes in and out without much control, which isn't very good for a cell. And if you're impermeable, nothing gets through, which isn't very good for a cell. But in between that is selectively, let me add the "ly" there, selectively permeable, which means the cell can decide in a way or it can control the things that move in and out. The other thing cells do, or cell membranes can do, is they provide a means of movement. Provide a means, I'm having trouble writing, means of movement. The cell membrane is fluid. Remember, I'm going to put that down here. It's fluid, which means it can ooze and shift and change its shape. And so the cell can literally cause extensions of the membrane to reach out. If you think about an amoeba, if you've ever heard of an amoeba, the way it moves is by extending its membrane and kind of oozing along. So the membrane has the ability to do that. It's very flexible and fluid. It also identifies the cell. We talked about the little molecules on top of the membrane, the glycolipids and glycoproteins and the peripheral proteins and things that help identify the cell as being part of an organism, like in your own body. And of course, this is also the way that the cell interacts with its environment. Everything that's around the cell membrane is where it comes into contact with first. All right, there's the cell membrane. Now we'll move on to the nucleus. The nucleus, there's no particular order. The nucleus controls the cell, or all the activities of the cell. I.e. of the cell. And the way it does that is kind of special. Let me put a little arrow here. This is how it does it. It contains that ever important DNA molecule, or several DNA molecules, which is where we find our genes. Patterns on the DNA molecule form our genes. And then these genes are basically the instructions, which are the instructions for building protein molecules. So inside there's the DNA. The DNA contains patterns, which are called genes, and the genes are the instructions for building proteins. It's kind of a complex, multi-step process. Now remember that proteins, of all the molecules that we studied, are the most complex. They're very, very big. They're made of different chains that are all wrapped and bundled into like a blob of string, and they can change their shape and they can take on many, many roles. All right, so what the proteins do, let's say building proteins that control and regulate the biochemical reactions that are happening inside the cell, and basically those are for all cell processes. So everything a cell does is somehow related to some kind of chemical reaction that's happening inside, and the chemical reactions are controlled by the proteins. Remember, when they do this, the proteins have a special name. They're called enzymes. Enzymes, which are like catalysts, right? They're catalysts that help control these regulations. So an enzyme is another word for a catalyst, except it's a special biology catalyst, okay? And those control everything. So your instructions are on the genes, the instructions build the proteins control the reactions, and the reactions control everything the cell does. So that's basically how that goes together. Okay, there's no particular order, so let's go with the cilia next. And we have the cilia, which serve as, I'm going to put their main purpose first. They're basically short. They're like hair-like structures. Of course, they have nothing to do with real hair. They're not made of the same thing. They just look like little tiny hairs, but they're not hair as such. They're structures, membranous structures, projections that come out of the membrane. hair-like structures that can vibrate or move. They can wiggle around. They vibrate and sway and wiggle. And they can do this in patterns. The cell can control which ones vibrate when. So they sway vibrate in patterns that allow or create currents. That can move the cell. Or move things outside the cell around. This is interesting. Move things outside the cell. Around. When you have a single cell that lives alone, the cilia quite often are important for movement. But when you have cells that live together in a big thing like your body, sometimes they use their cilia to move things through the body. So like when you have mucus in your throat, that mucus is, all the cells in your throat are sort of beating their cilia and pushing the mucus up into your upper throat, so when you can cough it up and spit it out or swallow it or get rid of it so it doesn't clog up your lungs. So we find cilia in places where we have to move things around in our bodies. Okay, so that's the cilia. Ribosome. Ribosome is the site where proteins are assembled. And it's quite interesting when you look at the actual mechanism of this, assembled. When you look at the actual mechanism of this, which you'll study in your grade 12 biology class, you're going to learn the details of this process. It is very much like a machine, like an assembly line machine that we would build like cars on or something. It's quite neat how nature actually mimics a lot of the things that we do. Or in fact, I guess you could say we mimic nature because nature started doing it first. So it's where proteins are assembled. So remember, the nucleus is where we have the instructions for the proteins, but those are just the instructions. And so there are messenger molecules that bring those instructions out of the nucleus into where the ribosomes are. Remember, the ribosomes were very close to the nucleus, right? They were on that windy thing, that endoplasmic reticulum. And we're going to see that the endoplasmic reticulum, we'll do that one next, is sort of like a tunnel or a channel where these messenger molecules can go, and it can lead them to the ribosomes, where then they bring those instructions, and then the ribosome begins to assemble the protein. So you could call the ribosome a protein factory, in a sense. That's what it is. It's a protein factory. And that's its primary role. All right, so let's do the endoplasmic reticulum next. The endoplasmic reticulum, the long, windy thing in the cell, is what it means. And again, the endoplasmic reticulum is actually very important for many processes. The endoplasmic reticulum, we often just call it the ER for short, because it's hard to say that big word. And the ER is actually a membrane structure. So it's very similar in its makeup to the cell membrane. It's basically got a bilayer of phospholipid molecules with proteins embedded in it, and all the characteristics of a membrane. The membrane that winds itself through the cell. Throughout the cell. So in our picture, remember, we only drew it in a few spots, but really it's all through the cell. And this membrane then forms channels for the flow of molecules. So we talked about how, molecules. We talked about how the DNA instructions have to get written onto a messenger molecule, which then leaves the nucleus And it travels down the endoplasmic reticulum until it finds a ribosome, which are those little dots we drew on it, so that those instructions can be delivered to those factories where the proteins can be made. The other thing about membranes are very important chemical reaction sites. A lot of the biochemical reactions that happen in a cell are associated with or happen with a membrane, chemical reaction sites. And there's two different ways this happens. Don't forget, the membrane has lots of molecules all over it that other molecules can interact with. The protein molecules can do all kinds of interesting things with chemical reactions. They can catalyze them. They can make them go faster as enzymes. They can bring molecules together so that they're more easily joined or broken apart. And the other thing that membranes do, so chemical reaction sites, and I'm going to put concentration gradient. Concentration gradients. So there's sites for concentration gradients. So what that means is this, just so you understand that. If you have something like this and it's liquid and it's got lots of stuff in it, the stuff will just kind of spread out all through the cup. But if you take a cup and you put a membrane down the middle, like the endoplasmic reticulum, then what can happen is things can build up on one side and there are not very many on the other. Right? It's a separation of these things. Now the membrane can select how these things move through. But what happens is if you get a lot of stuff on one side and not on the other, there's sort of what we call, like, I guess you could say it's like a pressure. These molecules would like to even out the solution. They would like to sort of push their way over here, but the membrane is holding them back. And that creates a kind of a pressure. And that pressure is called a gradient. That's what we call it, a concentration gradient. So that's a big word that you'll learn to use a lot more often in your senior years, concentration gradient. But it's basically just describes something like that, where the membrane is holding molecules back so that they're all accumulating on one side only. And then what can happen is, as little gates in the membrane open and close, they can be allowed to move and do their thing. And this is a very important process for many chemical reactions that we'll get to later. Okay, so there's the ER, the endoplasmic reticulum. Let's do the mitochondrion next. So one is called the mitochondrion. Okay, but the plural, if we want to talk about many of them, is mitochondria. So you don't look and say, hey, look, there's a mitochondria. You say, there's a mitochondrion. But if there's a crowd of them, you say, there's a bunch of mitochondria. And of course, those were the little, I think they were purple in our picture, and they're interesting because they're a double membrane structure. Now, what's neat is you're going to see an example of what we just talked about before. The double membrane structure means they have an outer membrane, and then they have another membrane inside that winds around like this. And so there's a space between the membranes. And then what these membranes do is, if I just go back up here, just like the endoplasmic reticulum is a membrane, these membranes create concentration gradients and differences in where the molecules are. And when they do that, that results in all kinds of very interesting movement of chemicals and molecules, which helps the reactions happen that allows the mitochondria to do its main job, which is to, it's where the combustion of glucose occurs. So in other words, this is how we generate. I'm not going to put power. Power is not really what it generates. What it generates is energy. There's a slight difference in those two things. We often say powerhouse, right? But really it's the energy house. It's the energy house, or the energy production for the cell. That's its role. And of course, the way it does that is by using the famous equation where it takes glucose, C6H12O6, and we get that through eating, right? And then it adds to that 6 molecules of oxygen gas, which we get through breathing, and then it converts those in a very complicated process into 6 carbon dioxide molecules and 6 water molecules. And in the process, it releases energy. Now, the energy that's released gets locked up into another molecule in the process. And you probably don't know much about this guy yet. The molecule is called ATP, which stands for adenosine triphosphate. That's actually all one word, triphosphate. That's all one word there. Adenosine triphosphate. It's a molecule called adenosine, and it has 3 phosphates stuck to it, so that's how it gets its name. It's a complicated molecule, and you'll learn all about that in your grade 11 and 12 biology as well. But for now, if we just remember ATP, whenever you see or hear about that, that's how a cell basically stores the energy that it gets from glucose. It makes these molecules, and these molecules hold on to the energy because it's found in the bonds between the phosphates. These phosphates, 3 of them are stuck on there. And if you pop them off, then whatever was holding them, the bond that was holding them, is energy that can be used for something else. And we call this, of course, this entire process, we call it cellular respiration. Okay, respiration. And remember, when we think about respiration, we think about breathing. Well, that's only part of this actual process. Breathing is the oxygen, but eating, or if you happen to be a plant, you can use photosynthesis to make your own glucose. You don't have to eat anything else. So cellular respiration refers to this entire process, right? And remember that photosynthesis is really just the backwards process of this, and we'll talk about that when we start getting into plant cells. So that's a very important reaction that happens, and it's a combustion reaction because it's a fuel combined with oxygen, and it produces carbon dioxide and water. Okay, so that's our mitochondrion. Next, we will talk about the vacuole. The vacuole. Now, the problem is this word is really just sort of a general word, right? So it's a membrane-bound space or bubble or pocket inside the cell containing water and other dissolved molecules. But the vacuoles have many different sort of purposes. So when you look at a cell, I mean, I'll just draw a quick picture here. We drew our cell and we drew a little vacuole. It looks like that. Well, that doesn't give us much, right? And we talked about, when we were drawing it, how different vacuoles will have different things inside of them. And so they will have special names if they perform a special role. So a vacuole, can be called a vacuole in general, right, for any kind of thing in the cell. But there are some that have certain special jobs. And so we talked about the lysosome, which is like a vacuole. It's exactly the same thing. It's a membrane-bound space. But it contains very special things. It contains digestive enzymes. And you know what those are? Those are protein molecules that can break down other molecules or break apart other molecules. Digestion is the breaking apart of things. So when we eat a banana, it goes into our system and we have to break it down. We have to smush it up and crunch it up and grind it up. But then deep inside our stomach and our intestines, enzymes go in there and they start to break apart the actual molecules inside the banana. They digest it. So when you're done, there's no more banana. It's like all the pieces of Lego. You built this nice Lego spaceship, and then if you pull all the Lego pieces apart, it's just now a pile of Lego that can be used to build another thing. And so digestive enzymes. So a lysosome, the word lyse in biology means to pull apart or break apart. So you can remember that. Lysosome. A lysosome is digestive. That's one example. There's another example that we can do, and it's called a peroxisome. So it's very much like a vacuole. If you looked at them under the microscope, they'd probably look identical to each other because they are the same sort of structure. But the peroxisome contains different enzymes. It contains enzymes to break down peroxide molecules, hydrogen peroxide and different peroxides. I'll put peroxides, which are toxic to cells and poison. So this cell, its job would be if the cell encounters peroxides, and peroxides can be formed by different processes, so they're popping up now and then, and the cell has to get rid of them or break them apart before they do any damage. So a peroxisome. And essentially, you'll notice they all end with zome, peroxisome, lysosome, and so on. And they're all sort of the same as a vacuole. There are other vacuoles, though. Other vacuoles, as we'll see in the plant cell, sometimes it's just a vacuole that controls water movement or water movement. So it basically helps the cell, let me respell that, helps the cell stay with the appropriate amount of water. If it can store water, right? And so if the cell is getting too dry, the vacuole can just release some of its water back into the cell to rehydrate it. Or if the cell is too wet, it can expand and hold more of the water. It can control water, and what's really cool is in some cells, not only can it control water, but some can contract and squirt water out of the cell. And if they do this, you can squirt water out like a little jet, which can cause your cell to shoot forward, just like a little jet plane. So sometimes we find those are called contractile vacuoles. So the point I'm making here is that these vacuoles are little pockets of membrane that serve all kinds of purposes in the cell, right? All kinds of purposes, and there's many different types and kinds, but there's some good examples for us, and that's probably enough for that. All right. The flagellum, we'll do the flagellum next. So one flagellum, or the plural, is flagella, just like the mitochondria, flagella. All right. It's a whip-like structure, sort of like a tail, I guess you could say. And what it can do is it can spiral around like a little propeller, or it can reach out and sort of pull things through the water. And so a whip-like structure that provides, or that moves the cell from place to place. So locomotion, that's what it does, moves the cell around. Lots of cells have flagella. We say they are flagellated cells. The most common one, of course, is the sperm cell. Everybody knows a little sperm has the little tail that wiggles out the back. That's its flagellum. But some cells, especially bacteria cells, they have many flagella. They have much more than just one or two. They can have 8 or 10 or 12 or whatever. And these all sort of work in conjunction with each other. It's complicated, right? It's kind of like, thinking about a spider having to coordinate 8 legs when it walks. But nonetheless, flagella are essentially whip-like structures, and therefore moving the cell around. Okay. And they are actually quite interesting when you study their details. They have a really cool little motor system that they run on, and it's very much like a little motor, just like an electric motor when we look at it under the microscope and study it. Okay. All right, we did the nucleus, but we did not do the nucleolus. So let's do the nucleolus, which sort of means the little nucleus. And it's found in the nucleus. It's a little dense area, or a dense region in the nucleus. And its main job is very simple. It makes ribosomes. So those ribosomes, which are the protein factories, well, guess what? They have to be built somewhere too. And so they are made in the nucleolus. We'll just leave it at that for our introductory thing. All right. The other one, I think, that we had on our picture, we had centrioles. The 2 centrioles. The centrioles are, remember, they were that little structure that they kind of look like a little cross when they sit together. They're like fibrous, weird-looking things in the microscope like that. The centrioles are for, they help organize cell division. Cell division. In other words, at some point, a cell has to grow and is split into 2. That's how cells reproduce. They don't get pregnant and have little baby cells. Instead, they just grow big, and then suddenly at some point, they split into 2 and become 2 cells. And we'll study that process very soon. And these centrioles are very important in organizing that. And so we'll see how that works when we get to that process and we study it. These centrioles, essentially, they separate and they go to the top and bottom of the cell, and then they form an anchoring site for things. We'll talk about the cytoskeleton shortly too, and the little they just make the whole process happen. So we'll study that later. No point getting into those details just yet. Let's do our next. I think the last one on our picture, last structure, was the Golgi Apparatus, or the Golgi body apparatus, Golgi structure, named after the guy who sort of invented them. What these are, they're little membranous, so they're made of membrane also, membranous stacks. They look like little stacks of pancakes. I'm going to put in brackets to remind you if you see them. They look like pancakes in a stack. And they're actually, they're on the endoplasmic reticulum. In fact, they're probably extensions of the endoplasmic reticulum membrane, just kind of wrapped up. But they're sections of it that do a very special job. So remember what happens in the cell. You've got the DNA instructions in the nucleus. You've got a little messenger that runs those instructions down the ER to the ribosome. And then the ribosome is where the proteins are made. Well, then the proteins, after they're made, they continue down the ER until they bump into these little sacs at the ends or near the edges. And they are then sort of prepared for whatever role they're going to do. And so by prepared, what I mean is you take a basic protein, but maybe you need to add some little molecule to it. or you need to change its shape a little bit to make it active, or you have to do something to the protein to get it to do its job. So it's kind of like, you know, the protein is made by the ribosome, but he just kind of like not ready yet to do the job it needs to do. So there's some processing that the cell has to do to adjust it and tweak it to make it ready. And so that's what's happening here. Let's put processing and preparing newly made proteins for their jobs or their roles. And that's probably all we need to say about the Golgi apparatus. The only other thing on our picture that I noticed, again, was the cytosol. The cytosol is basically water-based solution. It's a water-based solution containing many organic molecules. So organic molecules are molecules made of carbon, and they're all those things we talked about. It's basically, it contains, and it's just floating around, teeming with all the different Legos waiting to be used by the cell to build all the different things. You want to build a ribosome? Well, where do you get the bricks? Well, the cytosol has all these molecules floating around, waiting to be grabbed and used. Many organic molecules, and also minerals. I'm going to put salts and minerals. Technically, minerals includes salts, but whatever. We'll write them both. Salts and minerals for all cell processes. So you can kind of think about this as the, the App Store. It's where you go and buy stuff that you need, and where you get everything you need to build and make and do all the things cells do. It's this constant sort of warehouse of basic building block molecules that are floating all through the cell, the cytosol. Okay, and I think that covers everything. I will go back and make a point that I forgot to make, and it was about the centrioles. I'm just going to add to this. For some reason, we only tend to see centrioles in animal cells. Our next video is going to be the plant cell, and we're going to see that there are some major differences between plant cells and animal cells. And one of them is that the centrioles, we don't really see those in plant cells. And nobody's really sure why. It's just kind of one of the weird differences that happened way back in the evolution of things. All right, and there we go. So that finishes the animal cell and all of its components and all of the things that are in it and the things that it does.